My name is Christian Picard, and I'm a senior research biologist at Smithers, and I'm here to give a short overview of a poster that will be presented at the virtual CTAC Europe conference in May. The poster is entitled Historical Sensitivity of Common Plant Species Used in Non-Target Terrestrial Plant Testing. So, non-target terrestrial plant testing typically looks at the effects of plant protection products on seedling emergence and vegetative vigor. These test types have OECD guidance and analogous EPA guidance. Um, and depending on the regulatory objective, these studies often require testing up to 10 common plant species. And within the test design, there are a number of lethal and sublethal endpoints that are measured. And the studies often require generating no effect levels as well as various ECX values for use in a regulatory framework. So the size of the data sets make these tests very challenging. There also is other challenges such as the frequency of shallow dose responses, hormesis, and lack of standardization in the test methods. So in this poster, we wanted to look at our historical database and see if there were one or more species that were driving sensitivity in these tests, and could we use that information to um, design more intelligent tests moving forward. So we compiled endpoint data on 32 seedling emergence and 25 vegetative vigor studies. We assessed species sensitivity in terms of the lowest reported endpoint for each test type, as well as across compound classes and modes of action. So compound classes were defined as herbicides, insecticides, and fungicides. And then we mainly looked at mode of action within the herbicide class. And then we also assessed sublethal endpoint, minimum significant difference, or PMSD, for each species across the test types. So we looked at this statistical value to see if it was a indicator of true sensitivity. So a summary of the amount of data that we looked at roughly 1,600 ANOVA comparisons and around about 230 EC25 values. And in terms of the ANOVA comparisons, almost 1,400 resulted in no effect or no significant differences or no effects, which was roughly 84%. So these figures are a little challenging to see, but this is basically a frequency distribution comparing individual species sensitivities for seedling emergence and vegetative vigor data sets. So we have seedling emergence here, vegetative vigor. So whenever a species was determined to be the most sensitive for a particular endpoint, it got a score on the frequency chart. So essentially, tomato and bean are the most sensitive species here, and tomato and cucumber for the vegetative vigor. But in both cases, tomato is the most sensitive species. These are box and whisker plots of PMSD data for dry weight and length endpoints in both the seedling emergence and vegetative vigor studies. Again, a bit challenging to see here, but the take-home message is there was really no clear pattern that the PMSD data matched up with for the true sensitivity data. So tomato in many cases were kind of middle of the pack as far as PMSD, indicating that it took a larger percent inhibition for something to come up significantly different. So it wasn't really statistically sensitive, but maybe truly biologically sensitive to certain chemicals. Um, so some general conclusions here. Again, tomato was the most frequently generated, the lowest endpoint in both test designs. There are 84% of the ANOVA comparisons resulted in no effect outcomes, which is quite a bit. And the PMSD was not a clear indicator of true sensitivity, suggesting there was a difference between that statistical value and actually what was biologically sensitive. And not pictured here, but we didn't see any clear trends across the compound classes or the various modes of action. So moving forward, things like tomato and bean and cucumber, which tend to have more of a vine-like morphology, seem to be more sensitive. So if we were going into a test and had limited resources, those were maybe species of concern that you would want to add more replication or potentially more dose levels to determine of dose response. So if you have any questions on this, you can contact us at infoers@smithers.com. And thank you for your time.